Now I should skip this perhaps and go on to this problem of showing to you that if we would like to know the details of this complex metal cofactor, we need high resolution. Now, this is the 1.1 angstrom resolution which we actually obtained, but this is the more conventional 3 angstrom resolution which you get with protein crystals. You see the same cofactor looks like this, so a blurred, even broken electron density. There would be no way to differentiate the individual atoms in an electron density like this. So we very much need high resolution, and this we can get by a new technique which we developed in the last 5 years, namely a way to improve crystal quality. This was the initial crystal that we examined as it came out of the crystallization droplet, and you see blurred diffraction at high resolution. And now we start the protocol to reduce the humidity of these crystals. And you see what happens. So we vary the humidity gradually and look at the diffraction pattern. Now it disappears again, and it suddenly you see the high resolution diffraction appears, and then we dry out more, we lose diffraction again. Then we come to a plateau, almost have lost diffraction completely, and we go back to this optimal diffraction that we had found before and have now an almost perfect crystal with high resolution. Then we go back to the initial conditions where we found the crystals and we lose diffraction again. So high resolution is essential to do this kind of analysis. Well, let me come to the last part then of what I'm going to say, namely, now we do have the structure, so the molybdenum cofactor complex, we have identified this copper. How do we know how it carries out the enzymatic reaction? Now, for that purpose, we have to treat these crystals with inhibitors or with substrate analog inhibitors. Now, this is the identification of these various ligands. So there's the molybdenum, there is a copper, there is a bridging sulfur, there is a hydroxyl group, and there is an oxo group, and there is this so-called molybdopterin cofactor. So the first thing that we can do is to look at the oxidized form. That is, we have the crystal in air, and it is completely oxidized. The molybdenum is state 6, copper state 1. And then we can reduce it simply using an excess of CO. We mix in hydrogen, we mix in diisocyanate, a chemical reductant, simply to get 100% reduced state of the enzyme. Then we have molybdenum 4 and copper 1. How does that look like? And how does it compare? It changes very little if we compare this molybdenum 6 and the molybdenum 4 form. There is a longer distance between the molybdenum and the copper. Now this group, the molybdenum hydroxy group, has lengthened probably from an oxo to a hydroxo bond. This bond has become longer. So relatively little changes. So we went on and used inhibitors, substrate analog inhibitors. And this is the substrate, CO. Now we knew that cyanide, which is very similar to CO, inhibits, inactivates. And there are isocyanides, which also inhibit. And these we can study by crystallography again. If we use cyanide in large excess, then we find that this removes the copper. and we get an electron density that looks like this. And of course, this species is totally inactive. It has lost an essential part of the cofactor. The copper is lost. Now, this we can follow by measuring the activity. We also can follow the electron spin resonance and see, in fact, that the copper is lost, which gives a characteristic, together with the molybdenum, a characteristic electron spin resonance spectrum. And then we can bind N-butyl isocyanide to it. I come back to that later, perhaps, to introduce you to these various ligands. This is the CO substrate. This is the cyanide inhibitor, which leads to inactivation. And this is the butyl or butyl group isocyanide. How does that look like? You see, it becomes intercalated. You remember the free form of the enzyme had this molybdenum, sulfur, copper arrangement, and now the butyl isocyanide intercalates. And this is a strong bond formation between all the components, and this compound permanently inactivates the enzyme. So it binds like this. But we can take that as a model for the binding of the substrate. So it is a way to find out about dynamics of protein, enzyme reactions. Now we do work with crystals, and crystals are paradigms of rock, of solid objects. But what we do is we work like a photographer that finds out about the movement of the horse and the rider by simply taking snapshots, you see. Now 4x4 or 5x4 of them, and then if you project them fast enough, you see the horse moving. Now what we do is we incubate these protein crystals with inhibitors that show various views of the enzymatic reaction. And in that way, we are able to derive the dynamical aspects by protein crystallography of an enzymatic reaction. This is what is now shown here. So we have, as I said, the oxidized form, we have the reduced form, we have various inhibitor forms. This is the ground state, the free enzyme, molybdenum, sulfur, copper, and then we see what happens when we make a movie. So we have the experimental structures of individual states, and then we combine these informations to make that movie, and to see to show to you that there is substantial motion going on during the enzymatic reaction, which we can analyze in crystals by a process which I have shown to you. So this is the enzymology now. We have CO bound intercalating between the copper and the molybdenum. So this is activation of the CO by the copper, the transition metal. We have the molybdenum providing the polarized, the molybdenum providing the polarized hydroxy group, and this is the product which is formed when, which then can move away and the reaction being completed. By this process, MO is reduced to molybdenum-4, and the electrons are transferred through this cofactor via to the flavin via these 2 iron 2 sulfur clusters. Well, I should stop here, I think, and acknowledge the collaborators. It was relatively recent work, the last 3 or 4 years, collaboration with the University of Bayreuth, with a biotech company who helped us improving the crystals, with the synchrotron people in Hamburg, and the collaborators, my collaborators at the Max Planck Institute for Biochemistry. I would like to thank them and you for your attention, I hope. Thank you.
Thank you, Professor Huber. It's one of the lectures where I felt completely illiterate, but I think try to make something. This is, we'll open up for some questions. And Professor Huber has said that you can ask questions outside the topic, because the main theme being science and peace, so you could be asking questions in a broader range of subjects. But I think I have few of my colleagues whom I thought are knowledgeable in this area, so maybe they will inquire something. Yes, Professor Chongrak.
Professor Huber, my name is Chongrak Popasert. I am Dean of the School of Environment Resource Development. Thank you very much for the very impressive presentation. As President says, we scientists are always excited to meet with the Nobel Prize laureate. And today your lecture easily gives us a lot of inspiration of how to do research, how to search for knowledge, and how to apply knowledge into practice. Now, our field is on environmental engineering, and we are very concerned with the pollution caused by carbon monoxide. We know that carbon monoxide, CO, are generated by automobile exhaust, coal-fired power plants, and so on. And the main technology used to remove CO, like in automobile, is only catalytic converter, physical chemical process. From power plant, we use like scrubber, physical chemical process. Not much using technology, the carbon monoxide bacteria that you just mentioned. And today we learned that either aerobic or anaerobic bacteria can be grown and used to oxidize CO. Can you comment whether can we apply this CO bacteria like in automobile exhaust to oxidize them, or even to produce hydrogen gas, which is we call clean fuel? What are you thinking, or what are the current status of technology of using this CO bacteria in pollution control, for example? Thank you.
Well, thank you. Now, I don't think that we can use this bacteria in automobile exhaust. They just would be killed there. There is perhaps a way to use them in large biotechnological plants to produce dehydrogen. There is a certain possibility, although I would also doubt that such a process can compete with the technical process that is already available, the water-gas shift reaction. What we can do, we learn from looking at these structures. Perhaps we learn about making better catalysts, more efficient catalysts, that are stable without the protein environment. We still are unable to synthesize chemically these complex clusters. In the chemist's hands, this was still not possible, but perhaps there is a way to synthesize these and then perhaps have better catalysts which can be applied for in a technological process. Now the bacteria probably can't. This morning I was asked a related question about photosynthesis. Can we use what we see in the biological photosynthesis, which is extremely efficient, you see, compared to the technical photocells? Can we apply that? And I gave the same answer. It would not make sense to make photocells from proteins, which are very sensitive to light, to oxygenation, destruction, bacterial, degraded by bacteria and so on, so forth. But we can learn from these structures, perhaps, to make our technical devices better.
Good. Professor Stevens?
Professor Robber, first, many thanks for your beautiful presentation and especially the work you showed on that active enzymology, where you could see how the whole active site was really moving while acting and doing its catalytic action. Was very beautiful. I have a few questions, and I will limit myself to two. The first one is you showed that kind of corridor in the molecule for CO, that it can go into the dehydrogenase only in a rather specific way, but there is space for the molecule to move to inside. But I wonder, when the CO is such a polar molecule with a minus and a plus side, can it pass through the interior of a protein fast enough to enable the system to catalyze sufficient molecules per unit of time?
No, it obviously can. We can measure the turnover numbers. So what we cannot is visualize CO, how it travels to the active site. We know that the active site is deeply buried inside, and somehow CO has to reach. And the picture I have drawn is that there is a protein-free tunnel, you see, from the outside to this active site. And we suggest that this is used. We do not know for sure. Now, I wouldn't call CO a very polar molecule. It carries partial charges on the C and the O. And I think there are data around that show that small gaseous molecules quite easily penetrate proteins, even if there is no such protein-free channel that we find here. So I have no real problem from these data. You see, let me just remind you on the classical and very old data of oxygen diffusion into proteins as measured by their effect on fluorescence quenching of internal tryptophans. These are buried tryptophans. There is no tunnel, no channel to them, yet it is known and it has been measured that oxygen can penetrate, you see. And I think CO also in principle can penetrate, but it might be easier, it might be faster if there is a protein-free pathway. This is what we suggest. We can't see it
Could it be that some allosteric mechanism is involved?
Some?
Some allosteric, that the protein is opening?
No,
temporarily?
not really. I mean, that we would have seen in one of these inhibitor complexes. You see, I would think that we should have seen, when we compare the free enzyme, the reduced enzyme, the inhibitor-bound enzyme, that there should be structure changes. There are none. Except these small ones which I showed in the movie at the cofactor. There is motion, but not at the protein level.
If I may, Mr. Chairman, another very general question. Sometimes we, as biochemists, we can learn from the chemist, and we apply the organic and inorganic chemistry in biochemistry. But I wonder, in your beautiful work, it could be now that the organic chemist could learn from the arrangement of these methyl ions in the enzymes you have studied. And they might be able to, at least to do something to develop more and more efficient catalysts.
That's exactly what I believe too, and gave as an answer already to the previous question, that this is where we can help technical chemistry, showing them how nature does it, and perhaps there can be a mimic of this made. Yes, exactly.
Thank you, Professor Huber. Anybody else? Yes, Sadananda.
My name is Sadananda. I'm in computer science here. I'm not really very familiar with the technical details of the presentation, which is very interesting. But I'm interested in this area because it relates to some aspect of computing in which I'm interested, that is the cellular automata. Yesterday, I think you gave a talk elsewhere on the relationship between biology, physics, chemistry, and life, perhaps. So, as a layperson, I wish to hear from you, what is the meaning of life at the level in which you are talking, at the level of chemistry, at the level of molecular biology? I'd be interested. Thank you.
Yes. What is the meaning of life? Reproduction, for instance, is one of the key issues. And in case of animal life, it's movement. Now, plants don't have that, but they reproduce. Well, what we try to do now is first at the genomic level, analyze the hereditary material of living organisms, and we are extremely good at that. Even up to a human being, we know each of the nucleotide bases of its genome. Bacterial genome sequencing has become almost routine. Now, if there are probably 1 million or more bacterial species, there is still a lot to do. There are 500,000 insect species, and we just have the sequence of one of them, of Drosophila. So generations of scientists will be involved, but it is has become relatively automatic. Yet it's enormously rich in information that we get from genome, what we get from genome sequencing. In particular, let's say, if we so in the applied side, it is of great medical interest to look at the genomes of people that have an inherited disease and compare it with normal people, but at every other level as well, a virulent bacterium compared to a non-virulent variant, what are the differences, or what have you, or a salt-resistant rice with the wild-type rice, what are the differences, to identify the genes that are responsible for that. So this will bear enormously fruit to help medicine, pharmacology, crop science, and what have you. But this is at the genome level. Now the next level is the protein level, which is much, much more complex. Now, I often use the following picture when I speak about that. I show the photo of a butterfly larva and side by side with the butterfly. There is no relationship. If you wouldn't know that one is the larva of the butterfly, you wouldn't believe that. So they look completely different, they feed completely different, they move completely different, yet they have the same genome, you see. Each base is identical. But the proteome, as it is called, is different in these, and that makes the differences that I just described. So it's quite clear that this is now what we have to analyze, not only with larvae and butterflies, but with human beings, diseased ones and healthy ones, with resistant crops, and the development of the proteome during during the lifetime of an organism. So, of course, this change in proteome is also a sign of life. I'm not sure whether this is a satisfactory answer to you, but I see it more from the practical point of view, that we are far away from understanding the molecular basis of life, the chemistry and the physics of life. On the other hand, I believe life is chemistry, and if it is chemistry, then it can be reduced even to physics, but extremely complex. And we are far from understanding that even at the molecular level.
Thank you very much. Anybody else? Any? Yes? Yes, you can use it. Yeah.
Hi, good afternoon. I am Mary, master student of Food Engineering and Bioprocess Technology. Carbon monoxide is toxic, right? But you have this in your presentation that there are microorganisms who can live in carbon monoxide. So in the near future, would it be possible that we know now that this gene that encodes this specific protein, this carbon CO dehydrogenase, we can encode this to like human beings and then higher organisms so that we could live with this pollution and all? Would it
no, terrible, a terrible idea to introduce a bacterial protein into human beings. Now, it wouldn't, if technically, I think you can do it, but I don't think it would function. You see, it's very tricky, even for the bacteria, to make these clusters. They can make the protein, but then they need the right metal, molybdenum, which is a very rare metal, nickel, which is also a rare metal, the right metal at the right place. So there are transport systems in these bacteria which ensure that the right metal is at the right place to be incorporated into the protein. There are even helper proteins that help to incorporate these metals into the respective proteins. So it wouldn't help just to introduce the dehydrogenase gene. You would have to introduce dozens or perhaps hundreds of other bacterial genes into a human being, into a human cell. And I'm quite sure what the human cell would do. It would throw these out immediately. Okay. So, the way...
or before they start to absorb carbon monoxide, they will take care of human being itself, the bacteria. Well, any other questions? Yes, please. I think we'll limit this as the last question.
Professor, thank you for your inspiring presentation. And may I ask you another confusing question, which links up to Professor Stevens' question and Professor Sadananda's question. Since we're talking about peace, I think I'd like to ask you about madness. Since what can be further away from peace, or say peace of minds and madness? Rather, I'd like to ask you a question about mad cow disease and those agents which cause mad cow disease, that disease which we know devastated shops in Europe recently, and a very interesting link up to your professional experience as an expert on proteins. And those agents which cause mad cow disease, we were surprised to know about them as proteins. And these days we are forced to believe in these prions or prions, which we call them now, as living organisms. So what is your attitude as an expert, as a world expert on proteins? What is your attitude towards prions or prions as living organisms?
No, I think it's quite well established right now that these prion diseases are due to the misfolding of a particular protein which occurs in a healthy state and in a disease state. And this disease state has the capability of reproducing itself. So it catalyzes its own formation. It catalyzes the transformation of healthy prion protein into diseased prion protein. And this disease state is the dangerous one. It precipitates around nerve cells and kills these nerve cells. That, I think, is the idea that it's not a living organism. It's a protein which is somewhat unusual because proteins usually form a single unique conformer. You see. Now the prion forms one conformer where it is soluble and healthy, and another conformer where it has a tendency to polymerize, and in that state kills nerve cells. It's, by the way, not so unusual. I mean, we know, meanwhile, a number of other proteins, quite unusual ones, that we can transform into a polymer state, forming insoluble aggregates. I don't think there is so much special in physical-chemical terms about the prion protein, and it's certainly nothing that lives on its own. It catalyzes its own transformation, which is nothing but, let's say, a crystallization.
Thank you, Professor Huber. I will request those who have questions, we will have a reception following it, and you can follow up personally with Professor Huber there. Professor Huber, you would like to take a minute and sit down?
Yes.
Okay. Now I'd like to request Professor Mario Tabukanan to present a vote of thanks.
Distinguished speaker, guests, colleagues, students, ladies and gentlemen. I should begin by saying that this lecture is a very special lecture of special significance to the cooperation between the International Peace Foundation and AIT, because this is the 8th and the last in the series of lectures which we began in November of 2003. Through the International Peace Foundation, the community here at AIT had the privilege of listening to lectures in varied fields, varied topics. 2 gave presentations on the connection of science and peace, 2 on international intervention and peace, 1 on social responsibility of corporations, 1 on physics that lead to the development of nanotechnology, 1 on climate change, the ozone hole, and now again, a very interesting topic which we believe would lead to developments of technologies for the benefit of mankind. We would like to thank Professor Huber for, first of all, saying good words about the AIT at the beginning of the lecture. He did not know about the AIT until the International Peace Foundation sent an invitation to Professor Huber to give a lecture at the AIT. And the President has managed to convince Professor Huber that the AIT is indeed a good institution of higher learning. We thank Professor Huber for reminding us, when I say us, it's not only ourselves listening, but also AIT itself, for making a link between peace and science and technology. Because when we look back 45 years ago, the founding fathers of AIT had that in mind, that through science and technology, higher education, they equal peace, building peace in the region. So we thank you for reaffirming, sharing our thinking and the founding fathers' thinking on link of science, technology, and peace. We thank Professor Huber for giving us insights into his works, his colleagues' works at the Max Planck Institute that lead to his award of the Nobel Prize. And when I took my last course in chemistry 36 years ago, and it was a very elementary chemistry, so I should not venture into making a summary of the presentation, but this I can tell you, which I believe you would share with me, that I think the contributions, the research works of Professor Huber on protein and protein crystallography, crystallization, and other related works, I think would lead to developments of technologies with applications possibly in environmental protection, possibly in this problem of climate change, in health, in medicine, and perhaps in agriculture. So the seminal research of Professor Huber, I think we all appreciate, and we wish you all the best and good luck with your works at the Max Planck Institute. And from all of us here, we are so grateful for the time you have spent and sharing with us your thoughts on the subject of carbon monoxide utilizing bacteria. So with those words, I would like to invite our President, Professor Jean-Louis Armand, to make a presentation, a small token of appreciation to Professor Huber for your very kind lecture at the AIT.
Testimony of appreciation, the German AIT friendship. It's 30 years.